Hey, it's Mr. Mabry, and we are going to go over 4 commonly used stem cells that are used for medicine and for research. All of this information I'm going to talk to you about is found from our friends at the Genetic Science Learning Center from the University of Utah. In fact, you really don't need to listen to me. You can just go, if you are one of my students or anyone else for that matter, and you can read about them. But I'm filling out this note page here in Microsoft Word to help focus my 8th grade students on what is most important. Students, just so you know, as we go along through this graphic organizer, you can treat each of these lines as one line each worth of text. This should be one whole page. So you can either fill this out with me as we go along, or you can pause the video and set up this grid and then track along with me. So your choice. So if you want to pause it, pause now. Here we go. We're going to talk about our somatic stem cells. You may remember from our introduction, these somatic stem cells are adult stem cells, as it says here in our notes. They're the stem cells all adults have, and the purpose for these stem cells is for the growth and repair of damaged parts of your body. So, for instance, you might have heard your liver can replenish itself. Your heart, to some degree, can repair tissue. Your gut constantly has to repair itself, especially the stomach with that acidic stomach lining. You've ever had a skin burn before, you know your skin repairs itself. Muscle gets bigger. Your red blood cells, every certain number of days, roughly a month for your red blood cells, you have to replenish them. So it's important for us to have somatic cells in our body throughout our life. And of course, as teenagers, you can appreciate the fact that you're constantly growing thanks to your somatic stem cells. Now for potential here, this paragraph is going to talk about the potential to use them for therapy. So like here it says bone marrow differentiate primarily into blood cells. So you might think, well, it's not that useful, and it's not for doing things other than make bone marrow, but it's really useful if your bone marrow is shot because of cancer and you've got to borrow somebody else's. So it does have limited use because they're already differentiated, but it is special because you can share with others if immunosuppressant drugs are DERGs. What's a DERG? Drugs are used. And as far as the ethics of sharing, very few people say that this is evil or bad. So we're going to say that somatic stem cells aren't very controversial. Now, let's scoot down and talk about embryonic stem cells. You'll remember from our introductory video, magic happens, sperm and egg meet, here they are becoming a baby, and then they're dividing, and at some point, when we have this blastocyst stage, what some researchers can do is you can suck the blastocyst out and put it inside a petri dish, and you can keep those little embryonic stem cells alive inside that petri dish indefinitely as long as you keep feeding it and giving it the right sort of environment to live in. And then if you put these stem cells in different conditions and you expose them to certain markers or cues, then these stem cells can become any cell type, because remember, these embryonic stem cells, if they would have continued to grow inside the blastocyst, would have become a baby. So, as far as definition, remember this is the egg plus sperm. Those are embryonic stem cells. The potential is great, exclamation point, because they are undifferentiated. Now, you still need to remember, if you are using these embryonic stem cells, then they don't come from you, that you would still need, if they are shared with, I'm just going to copy and paste what I have up here, because embryonic stem cells, unless they came from your body, still would have tags that your body would want to fight off. They would think, oh, these are bad guys, get rid of them. So you would still need to use immunosuppressant drugs, but they're different from here because these have limited potential and these have great potential. I'll even capitalize, whoa, great potential. However, the ethics of this are very controversial because there's a question, does an embryo, or the question is, when does life begin? Are embryos babies or just tissue? That's the crux of the question, and something we'll be coming back to time and time again in the next few days. But let me introduce you to something that was sort of a game changer just a few years ago during your lifetime. Here's a third type of stem cell called induced pluripotent stem cells. And to understand what an induced pluripotent stem cell is, let's take a look over here. So this is a little crazy. What we can do now is we can take your skin cells here, and if we treat them with reprogramming factors, which to my understanding tends to mean we stick a virus in there with certain genes and it activates at least 4 specific areas of the DNA inside these skin cells. They can sort of be programmed backwards to sort of forget that they're skin cells and become embryonic-like again. So I'll say that again. We reprogram them so they sort of forget their skin cells and they sort of go back, roll back the clock, and now they think, oh, I'm an embryo. I'm an embryonic cell again. I wonder who I'm going to be. And then after these few weeks pass and they've rolled back the clock and become stem cell-like, depending on which environmental cues you give them, they can then differentiate to different cell types, just like human embryonic stem cells. So for induced pluripotent stem cells, we're going to say they are reprogrammed skin cells. And skin cells, they're used because they're so easy to get. The potential, they are great because they're undifferentiated. What is special about them is there are no immunosuppressant drugs needed because they're your own cells. So that's what's so exciting about these. No embryos are harmed in the making of these cells, and they come from your own body if you were to use them, so you wouldn't need drugs to keep your immune system at bay when you use them for treatment. And currently, the ethics, what they say here for ethics is, the same ethical procedure as all medicine. So what that really means, it is not controversial at this time to use induced pluripotent stem cells. Now this takes us to our last and final cell type I want to talk about, and that is, as you can see on our paper, it says embryonic stem cells made via therapeutic cloning. So here's the deal with therapeutic cloning. We're still making embryonic stem cells, but we're not making embryonic stem cells like we were over here, where a spermie and a little egg were meeting up to make an embryo. Instead, what's happening is a patient is donating some skin cells, and the nucleus is taken from these skin cells and inserted into a woman's egg. Now this egg has been denucleated, it's been removed. So in other words, this egg is just sort of a host for this person's nucleus. And once the nucleus from this patient is put inside this woman's egg, it's put in a petri dish and it's being initiated to grow and divide and to form embryonic stem cells inside of a petri dish that then, just like other human embryonic cells, can differentiate into any cell type. So what's unique about this is we're still making an embryo, but instead of using a sperm and egg, we're using the DNA from someone's skin cells and putting it inside a woman's egg. I know it sounds sort of far out and science fiction-y, but we've already been doing this for a few years. So let's go ahead and fill out our notes on this. So what we're doing here is we are making embryonic stem cells via something called SCNT, which we're going to learn about when we learn about cloning, for therapeutic purposes. This means for treatment of different things. Now the potential here is just like all the other embryonic and iPS cells. They're great because it's undifferentiated. And similar to iPS cells, no immunosuppressant drugs are needed to use therapeutic cloning cells because they're your skin cells and the DNA from your skin cells. And so it looks and feels just like your cells. But the issue is, you are making an embryo. So it's the same issue here. It's, when does life begin? Are embryos babies or just tissue? We just made an embryo, and now we're using this embryo for treatment. And not only are we making an embryo, but you are making an exact copy of you. This is your DNA. So you're not just making a random embryo, you're making a clone of you, an embryo clone. So if this embryo would have been put inside a woman, then that would have been a clone of you being born, which gives people the willies. So this here is very controversial for a couple of reasons. Number one, are we, quote, killing embryos for treatment? And number two, you're making a human clone, so to speak. It's just cells in a dish, but remember, if these cells are put inside of a person or a woman, a surrogate mom, then it would become a human clone. So, wow. There you go, folks. Those are our 4 different types of stem cells that we'll be talking about the remainder of this week. I think it's best when we have this graphic organizer to go ahead and do a little comparison contrast. So the last thing we're going to do is we're going to do what I call a strike meter. And we're going to go through and see which ones have strikes against them. So let's see, growth and repairs, fine, limited use. Okay, so this is going to be a strike, a big red letter. I'm going to do it in red. I encourage you to do it in color. So this is strike. And then, we need immunosuppressant drugs. That's another strike. So this guy, his strike meter, he has 2 strikes. 2 strikes against you. Sorry, somatic cells. Better luck next time. Okay, embryonic stem cells. Hooray! Great potential, good. Up, strike. Let's just cut and paste this. And oh, nope. Controversial. That's a strike. So just like somatic cells, you have 2 strikes against you, embryonic stem cells. Let's jump down here to therapeutic cloning. Hey, they are undifferentiated. Great. No immunosuppressant drugs. This is great. Oh, wait a second. People are very nervous about you. That is definitely a strike. So you have 1 strike, but that leads us to Mr. Mabry's very favorite iPS cells. Great potential. No immunosuppressant drugs needed. No one's given it a lot of controversy, so no strikes for you. You are winner, winner, chicken dinner. Good work, iPS cells. Woo! And we'll highlight you in red. Now forget red. You're going to get big boy blue. So there you go, folks. Those are our 4 cell types. Those are your comparison contrast of them. Make sure you know the ins and outs of the different types. So you are ready to enter a debate about how you feel about each cell type and especially how it relates to cloning. Thanks for watching.